Atwas wanted to come to the podium, and Professor Sharon Rosette from Bioinformatics. We are now open for questions and discussion. So, just a minute. Let those people who are not would not like to follow the discussion and. I think we can start. Okay, please.
Compared to the total population of these people. And so maybe how do you eliminate biases that are due to just sparse sampling?
So if you refer, say, to that list where I say there are like 30,000 changes, yes. So those are then changes that are present in all humans, and we have thousands of humans from all over the world today, so we know that these things are at very high frequency, 99% or almost fixed in humans. Even if we have a single Neanderthal, now we have more Neanderthals, but even if we have a single Neanderthal and it doesn't carry that change, it means that that change may have been there in the common ancestor of Neanderthals and modern humans, but it was sort of for surely not fixed at that point. It has sort of risen to high frequency recently in human history. Only when we have more Neanderthals would we really know that none of them have it, but I think that class of changes are still something that's risen to high frequency recent because we have so many present-day humans.
I'm sorry, maybe I missed it, but just a biological question. How can you extract a biological molecule as DNA to sequence it from a fossil?
Yes, so these are not fossils. I hope I didn't say fossils too many times. I try to avoid the word because, of course, if you with fossilization mean replacement by minerals, there will be nothing there, right? So these are not fossilized, the bones we study are not fossilized in that sense. They might maximally be partially replaced by minerals, but there are still organic molecules there. There's collagen in most of the things we study and DNA.
And how far back in time can you still search these biological, not mineralized, yes. I mean, can you go back to Homo erectus or even farther back?
So the oldest DNA one has retrieved is from a lab in Copenhagen that is studied in permafrost. This is horse remains that are 700,000 years approximately. But that's then things that have been continuously frozen over that time. So outside the permafrost, the oldest DNA we have is from our lab and from hominin remains in Atapuerca and Cima de los Huesos, which is more than 400,000, 430, 450,000 years. That's really a rare exception, that site. We have looked at several other sites in that age range and have not been successful, but under good conditions. And we cannot really say what good conditions are. We don't fully understand why that site is so unique compared to others. You can, yes, go back 500,000 years. Maybe we can imagine going back towards 1 million years sometime in the future. But we will for sure not get to, Australopithecus. We will not get to. It's my prediction, but I've been proven wrong before when I say negative things.
Thank you.
I have a question for both. Professor Papa, what is the origin of people today. I'm diabetics. That's why I'm asking about. You said that diabetics is the remain of the Neanderthals. If I understood right?
I didn't get that. Who is it?
Diabetics.
So this is just one genetic variant that contributes to type 2 diabetes. There are several other variants, but it is one that is quite frequent in Asia. And so say 25% of people in China carry this variant that predisposes them for type 2 diabetes if they also carry other variants and have environmental conditions that lead to this.
Okay.
So it is just this tiny part of the genome that those people carry from Neanderthals. It's not that if you have diabetes, you're more Neanderthal overall in some sense than anyone else.
Okay. But it's very interesting. Okay. Professor Reich, you talk about that the movement between Eastern Europe or the Eastern Eurasia and Western Europe is continuously. We know about the story in the Bible or in the other of the Philistines that came from Crete to this place. What do we know about them? Where are the ancestors today?
I don't have access to any DNA from the Philistines, and I think the history of this part of the world is very complicated for the reasons that you all understand, which is that it's been a corridor of movement of people, and I think it's not likely to be any less complicated in terms than European history, which we already know is very complicated, and we have much less DNA from more recent periods. But it's a solvable problem. So DNA from people like the Philistines or from cultural contexts associated with the Philistines, as well as other cultural contexts, connecting to people at different periods before, after, and today, could make it possible to learn about population movements. We simply don't have the data right now, but the data is eminently obtainable if one tries.
Okay.
I have a question here. Can you say now anything about the fate of the Neanderthals? Why did they disappear?
Not from the genetics, no. I mean, I think it's a reasonable suggestion that it has to do with modern humans. And I think I'm saying that because not only the Neanderthals disappeared, say the Denisovans disappeared, these hobbits in Indonesia disappeared, all other forms of humans disappeared when modern humans appear. The last, say, 30,000 years is sort of unique in our history in that we have no close relatives around. I do think it has to do with modern human behavior in some form, but it might have been different mechanisms in different parts of the world and so on. You know, in some sense, one could say if in the future we succeed in understanding what makes modern human sociality or cognition special, that may be the key to why these other groups disappeared. We will for sure not find the reason in the Neanderthal genome. You know, it will be in our genome that the problem is.
Yeah, I have a question here. Question to Svante. You showed very impressive data about the FOXP2. And the question is, it's a transcription factor. And I guess the downstream genes are well known. And the question is how the 2-point mutations affect downstream genes in the human mice.
So downstream genes are not that well known as you would imagine. There is some ChIP-seq data from cell lines, some downstream genes. What we are now doing is looking in the mice, actually, on single-cell transcriptomics from those parts of the brain and identify genes that are up and down regulated. And there are some interesting genes that actually change their splicing pattern. I would think that there will be alternative promoters and things that go on that will be of interest, but there's sort of no breakthrough there of understanding what happens.
Hi, thank you very much for your interesting talk. I actually want, it's a matter of terminology, maybe. You actually showed using ancient DNA that biologically Neanderthals and Denisovan are not a different species. So I can understand and relate why changing the traditionally Neanderthals as a different species is still there, but you found a new hominid, the Denisova, and you gave it, you still gave it identification of a different species. And if we...
Did I?
On Denisova, I don't know. People relate to it as a different species. Maybe you didn't mean to, but...
I mean, we try to judiciously stay away from taking a stand on those things, you know. If I'm a little sort of facetious, I would say, you know, defining what is a species here is a sterile academic exercise that gets professors excited, but no one else. It's sort of, if we show that, you know, yes, one can have fertile offspring, there are probably some barriers to this that are hinted at by these deserts of ancestry and so on, that's the interesting thing. If you want to say that this is two species, that's fine with me. If you want to say these are subspecies, it's fine with me. We sort of really stay away from using a Latin name that would force us to take a stand on this. I tend to talk about different forms of humans or something like that, and not use the species word. You know, we all know that speciation is a gradual process. It's sort of just an academic exercise to draw a line in the sand there and say, I would stay away from it.
Hi. Hello. Oh, sorry, David, did you want to continue or can I ask a question? Okay. I actually have two questions, if it's all right. First, I agree with you that this nucleotide archaeology is a huge paradigm shift, which can answer one of the fundamental questions in the discipline of archaeology. And how often does ancient DNA correlate with material culture? You gave one example of corded ware, but do we see this in most cases, or does it give a different history that archaeologists wrote?
That's a great question, and I think that what's so exciting, I think, about the beaker culture phenomenon work that I try to talk about a little bit is that it's a case where it does not, or does partially. So I think that one of the, actually I'm going to back up a little bit, and one of the, when we were writing our paper in 2015 with a very large number of archaeologists, co-authors, just before submitting the paper, we had a very, very strong response from a number of the Central European archaeologists who said to us, what your work is doing is reviving the settlement archaeology of Gustav Kossena and archaeologists from the beginning part of the 20th century that was used as the justification of scientific racism by the Nazis, where there was a conflation of material culture with people, and when pots change, people change, and that the ancient distribution of these people who would correlate with these pots would be associated somehow with claims to land and claims to expansion into certain areas. This was misused politically, and it's been anathema in Europe, especially after the war, to say that the spread of culture corresponds to the spread of genes. And I think that one of the things that the genetics has been doing has been being able to just test whether this cultures correspond to genes. And in cases like the corded ware or the Yamnia, or the linear band ceramic or the cardioware, there is a very strong correlation often between genes and spread of pots and spread of material artifacts. And so it's revived the idea of migration and mass movement of people in archaeology, which had been a subject of a lot of debate for dozens of years. And it's now possible to test, and geneticists are unschooled in this area. We haven't gone to graduate school in archaeology, and we're just making statements right and left that touch on these sensitive issues. So this is one of the things that's going on. And one of the things that I wanted to highlight with this beaker example is that, in fact, it's more complex. Of course, it's more complex. In the case of the beaker culture spread, the pot-making types, and it's clearly related, spreads from Iberia to Central Europe or perhaps in the other direction without any movement or minimal movement of people. It's the spread of idea like the spread of cell phone use or something like that doesn't necessarily involve movements of people. So it's both. But I think what the DNA is doing is it makes it possible for the first time in a long time to really evaluate empirically in a way that was not possible with morphology and anthropology and study of bone shape, whether people move. And I think this is something we have to grapple with, and it's an important issue. And so I think that we just have to deal with it. And I think this beaker example shows that it's complex. The beaker is not a simple example because in Iberia and Central Europe, it's very different. But the coming of the beaker culture to Britain is associated with a very dramatic movement of people, and it's correlated to the culture in that case. It's very complicated.
We have work to do. One more question. How do we see the future of ancient DNA in terms of new technology? Do you expect to see the graph continuing its linear acceleration, its exponential acceleration?
So I'm going to talk about the graph, and maybe Svante might comment about the technology. I think that we are at the beginning of the exponential increase, not at the asymptote. I think that within a couple of years, we'll have tens of thousands of ancient genome sequences. We will be able to routinely get DNA not just from West Eurasia, but from many other parts of the world, including regions that people have historically not thought was possible to get a lot of good DNA from because of innovations like the Petrus bone and the enrichment and improved techniques for obtaining DNA from marginal samples. And I think that their future is obtaining larger and larger sample sizes, being able to make richer and richer statements about relationships about populations, and really to grid sample across the world and really understand how ancient cultures relate to each other and populations today to estimate past population sizes, crashes, expansions, and all of these things, and really establish a temporal dimension to the movements of people that was until now really only possible to look at with the screen of present-day populations, which is really just a screen which hides everything that happened before. So it's a real transformative thing. And maybe Svante could comment about the technological curve, I think.
Yes, I don't know. I certainly believe that DNA work will become sort of standard in archaeology, that it will be a bit like carbon dating. You cannot imagine doing archaeology without this technology. I'm quite excited by this new possibility to look in sediments, for example, to retrieve DNA from the animals that have been in a bone and from the humans and hominins that have been. And I think that sort of it will also be extremely important to reconstruct the population history of the fauna, for example, from doing parallel studies in the future of the faunal remain and of the plants, etc. I think that all organisms that you find at a site is amenable to these types of techniques and will be studied. And yes, I think we will become more efficient. We're sort of now trying to design capture sort of enrichment techniques to look at the nuclear parts of the nuclear genome from sediments also, which is very problematic because you have all these other mammals there that you want to avoid and just fish out, say, the humans, but there might be parts of the genome that we can fish out and study. I think that one should, if I should make propaganda when you do excavations today, I would certainly advise to sample sediments quite intensely and just store for the future. You know, don't do anything but so that it's there, because it's frustrating with all these sites that have been excavated and you cannot go back to them.
Okay, so it strikes me that we academics also really like arrows that go in one direction and trees that are very clean and neat and sort of keep everything separate. And as a biologist, I've always said that's like chemistry, and biology always has cycles. And I'm wondering, it actually struck me that the work you did the work that you did shows that basically introgression is a very important part of all of the heredity that you're analyzing, and so my question really is, if populations end up not interbreeding but remaining very pure, shall we say, do they actually end up going extinct because of those problems? And is this really what we're seeing as an underlying thing here, which is that it's the movement and the bringing in of new genetic material, which keeps these populations vibrant and developing and adapting to their new and different surroundings.
So that's a very interesting question. And I think the question you're raising is the role of population mixture as an adaptive, the force for adaptation. And you could, so sex between men and women is actually evolutionarily advantageous. It combines traits in new ways to produce offspring, some of which are more adapted to the new situations they're in than their parents, and is a powerful force for adaptation to new environments, and perhaps mixing of populations that have been long separated could be that as well. I think there's no good evidence right now that mixing of populations has been an important force for adaptation. I'm certainly extremely interested in that possibility, and there are specific examples like the high altitude adaptation and perhaps keratin filament biology associated with Neanderthals and some others. But broadly, I have no evidence that it's been essential in any way to adaptation, although this is of high interest, because it's becoming increasingly clear that hybridization is an important force in evolution of species, and mixture of human populations, for example, occurs again and again and again over time, with it being very rare that you have long isolated populations. So to think what the biological consequence of that process is an interesting thing to do, but it's an open question how adaptive it is.
I mean, I would just sort of also like to a bit just warn against drawing sort of inferences of what is good and bad from the type of genetic work we do. I would certainly think that an isolated population can do perfectly well in where have you, New Zealand or wherever you go. There are examples of mammals, such as, say, beavers in Europe that were almost extinct and are like inbred mice today, but do extremely well and are actually a pest. So you can find individual genetic variants that have been advantageous, but I would imagine that modern humans would have done totally okay without mixing with the Neanderthals. In some sense, we study history here, and I would not infer any sort of ethical commandments of you should mix or you should not mix or anything like that. I sort of be very afraid of that.
Well, thank you for the interesting talks. And a question maybe to Professor Pablo and Professor Reich. Homo erectus was mentioned, and how about this Homo florensis actually? Where do they belong? Where is the admixture of these guys with some other species? Maybe it's a future Nature article, or maybe it's already under embargo, so I'm sorry about that.
Unfortunately not. Unfortunately not. We have tried, we are actually systematically trying animal remains from this site where the Hobbit is found on Flores. So far, I can perhaps say, is this webcast in the world or not? Well, I can say that, say, Holocene animal remains do yield DNA from there, older things so far not, but we are struggling with it, and if we can get the animal remains to work, then one might be able to study the hominins from there. But we really don't know. There is some interesting indication in the Denisovan genome that they have an old component in their genome that's not there in Neanderthals, that seems to come in from something that diverged more than 1 million years from the human ago in the human lineage. It's very tempting to say that that's some Homo erectus-like thing or something like that, but we don't know.
We see that the evolution takes a lot of time in order to change people or other animals. So we are now in a new time that machines will take our place in every aspect of life. Maybe it take 100 years, 200 years, 1,000 years, I don't know. But the behavior of the people and the living of the people will be in other form, or organic or something else. If this situation will happen a long time, if people will change, like the history before, another way. It's a little science fiction.
So different parts to that question, I guess. One is sort of that, yes, one can attempt to use, say, particularly at the moment, the mitochondrial genome to approximately date when some individual lived. It is still very approximate, say, and unknown error bars around that. So perhaps another part of the question to say when we date divergences between populations or between genomes, how accurate that is, and that is subject to uncertainties about the mutation rate in humans actually, which is about a twofold difference in opinions about that. That on the one hand comes from paleontology actually, that gives divergence dates between species and then we compare the genomes, and from new data about sequencing human families and seeing mutations in the children that's not near in the parents that tend to come up with much slower mutation rates that makes things older when you look at it. And I think ancient DNA is beginning to contribute to this. When you look at early modern humans and compare their genome to present-day people, you will again tend to come up with slower rates, more like the family rates. But anything we say, I have the feeling, anything I say in absolute years, I would take with big hesitation, but sort of relative dates, sort of if I say that something diverged 4 times earlier than that, that's something I would believe in. But what that is in absolute years, I would be.
Just wanted to add a short comment to that. Yeah, it's very distressing to archaeologists who are used to this precision of radiocarbon dates to see the uncertainty around many of these genetic dates. Often radiocarbon dates are accurate to within hundreds of years and can be meaningfully interpreted that way, and the genetic dates are, I don't know, somewhere between 230,000 and 450,000 years ago or something like this is probably when the event occurred, and it's not adequate for a lot of interesting inference. But there are some exceptions to that. There's different clocks in genetics, and one good date that we have is we know something about when the Neanderthal interbreeding with modern humans occurred. We know very precisely that it occurred between about 50 and 60,000 years ago, and that's because the chunks of DNA in some of these old humans that we have DNA from and we have radiocarbon dates on are of the size that would be expected from chopping up from a fairly small number of generations prior to the date when they lived. And so we can tell from that that the interbreeding of Neanderthals into modern humans occurred between 50 to 60,000 years ago, and the interbreeding of Denisovans into the ancestors of New Guineans and other populations occurred about 12% more recently than that. And those are meaningful numbers that I think can constrain the spreading of people through Eurasia and interaction with these archaic humans. And for example, are suggestive, although this is not compelling, that the Upper Paleolithic, the stone tool technology, may very well have developed after the initial diversification of, for example, East Eurasians and West Eurasians, and be contributed only to West Eurasian populations and their relatives among the ancient North Eurasians, which could perhaps be related to the funny distribution of Upper Paleolithic stone tools, which is West Eurasian and Northern Eurasia, but not Southeast Asia and Australia.
I just had a question about Africa that maybe connects it to talks. Because in Professor Pablo's talk, we heard about how most humans have some Neanderthal DNA, except Africans. So everyone after the exit got some DNA, Neanderthal DNA, because they mixed probably somewhere around here, the Middle East, after they exited Africa. So does that mean that there was no return migration at some point to Africa from Eurasia? And is that right? Because it seems strange to me, with all these movements that we heard about in the second talk, with Europeans more closely related to Native Americans than to other groups, it seems strange that for all this time there was no big back migration to Africa, and they also didn't get a piece of Neanderthal DNA. How does that work? What do we know about that?
I think that there's very substantial impact of non-African ancestry or non-African related ancestry in many populations in sub-Saharan Africa in different proportions. And it's clearly secondarily derived from migrations, almost certainly from the Near East, associated with the spread of farming and pastoralism after the Neolithic Revolution. It's especially common in Ethiopia and in Northeast Africa. It's even reached the far south of Africa, including in Khoisan Bushman populations. There are perhaps some groups that haven't been affected by it, especially in Western Africa and Central Africa. But I think that these are clearly secondary movements where the proportion of West Eurasian or non-African related ancestry tracks in a pretty proportional way to Neanderthal ancestry. So you do see bits of Neanderthal ancestry in these groups, and it's proportional to that proportion of their ancestry that comes from these lineages.
Hello, David. This is a question about the data that you're using, these 1 million variant sites. So do you think that moving on in the future, you might have to kind of reconsider if you want to get like more detailed inference or different types of kind of conclusions on migration events, that you might have to think about widening the set?
Yeah, the question is whether this set of million sites that we've been looking at for the last 2 years should be updated. And the answer is, of course, yes. There is a large effort associated with updating them because there's thousands of samples that have already been screened and will need to be returned to update the database. So I think that at some point that will become necessary for some analyses. But right now, a lot of the questions that we can answer are still answerable with these 1,000 sites, although it's not perfect. But definitely we can create chemical reagents that will assay other positions. And at some point, this particular technology we're using is going to be outdated, and so we move to something better. But right now, it's still an effective way of screening the whole genome.
And is there any way of figuring out how much that may be influencing like current inferences, or?
Yes.
Yeah.
I mean, for example, a lot of data generated by us and by other laboratories doesn't use that approach, and we could compare the inferences. I think that one example of how powerful this type of data is we have a pretty formidable group of population genetic analysts in our laboratory, and every few weeks we sit around the table and I ask people, we have all this shotgun data, which is whole genome sequencing data from a subset of the individuals in our study. Does anybody want to analyze it? And they all kind of try to look away because they just want to analyze the data from these much larger number of individuals processed this way. So people are trying to see what information they can get out. And there's very, very little information with current techniques. It's not to say that other techniques will make it possible to get other information out of whole genome sequence data, but there's very little information about history or even about biology that is relevant to the samples that we're looking at that could not be obtained with these million sites, and that's reflected in a diversity of analysts continuously confronted with the choice of what type of data to analyze. The large sample size is very powerful.
Well, of course, the inferences we make, we hope that other samples will not change this totally. But yes, it's true, things are melting, the permafrost is melting, and one could hope that things come out. So we've done a very informal effort with our colleagues in Russia. There are people who collect mammoth ivory. You may know that the price of mammoth ivory have increased dramatically since the ban on trade in elephant ivory. So there are people who in the fall travel with ships along the meandering rivers of Siberia and collect mammoth ivory. To sort of put out the word that we would be interested in human bones that they should find, and they should collect them, and we would even pay a bit for them. So we got a number of bones that have turned out to be very uninteresting, but that's how we got our hands on this 45,000-year-old individual from Ostishim that David mentioned, where we were able to sequence a high-quality genome, which is now the oldest genome of a modern human outside Africa and the Middle East that we have. But yes, one could hope that things come up. One could, in principle, even expect a Neanderthal to come out of the permafrost one day. But
one comment I want to make is that the data, it's almost impossible to imagine a higher quality genome than some of the ones that have been produced, for example, from the Neanderthal from Denisova Cave and the Denisovan from Denisova Cave. Even a permafrost specimen would probably not produce data that was much higher quality. So it's not as if you can expect a information content increase massively greater than what's possible even from non-frozen environments in some cases.
The mistake, they should have received the prize for the future dimension rather than the past dimension.
Next year.
Next year. Okay, if it is up to me, sure. And I would like to thank you all for coming today and being with us. And see you next year, as Svante said. Thank you.